Hello, everyone. Good morning, good afternoon, and good evening, depending on where you are. Thank you for joining this session. I'm Yang-Su from Microfluidizer, and I'll be presenting you the scalable microfluidizer technology for cancer drug delivering nanoparticles manufacturing today. Here's the agenda of today's presentation. I'd like to start with a quick introduction to tell you a little bit more about Microfluidizer as a company, then briefly talk about drug delivering nanoparticles and some common challenges and requirements during the development and production of those nanoparticles. And then talk about why the microfluidizer technology is a unique technology and ideal solution to address those challenges and requirements with a few case studies to show you the application of technology in fabricating drug delivering nanoparticles. Let me just bring my laser pointers real quick. I will end my presentation by showing you our diverse product portfolio and hopefully have some time for questions. Microfluidizer is under our parent company, IDEX. We're part of Material Processing Platform, or NPT, and there are the three IDEX segments, which is the Health and Science segment. Here you can see other brands under NPT platform. Microfluidizer itself was founded almost 40 years ago, and we specialize in design and producing high shear fluid processors using our unique interaction chamber technology. We're just located outside of Boston in Massachusetts and have a global presence and customers from all over the world. As I mentioned already, we're part of the IDEX NPT platform. Our technology serves many industries, primarily pharmaceutical and biotech industry, but we're also widely used in other industries, such as chemical, food, and nutraceuticals, as well as cosmetic, in both development stage and commercial scale manufacturing. With our extensive experience in applications as well as machine designs, we can provide tailored solution to enable you to achieve your specific processing goals. I think everyone knows very well that conventional drug delivery methods face a lot of challenges, including low solubility, which leads to low bioavailability, poor in vivo stability, and adverse side effects, etc. Nanotechnology is a fast developing area in recent years, which had led to the development and applications of a variety of nanoparticle-based delivery systems. Such systems offer many benefits comparing to the conventional large-sized materials. Just to mention a few here, nanoparticle delivery systems can achieve higher delivery efficiency with enhanced targeting effects. It can also greatly increase the bioavailability of the drug, even at reduced payload, and therefore reduce desired side effects. This is particularly important in cancer therapy, since most chemotherapeutics are highly potent and therefore their doses are usually limited during conventional treatment. So while you're working on the next exciting cancer drug or treatment, keep in mind that good delivery system can also help in many different ways. A few popular nanoparticles as drug delivery platforms are shown here as examples, and I'll be covering most of them later on with my case studies. When talking about developing and manufacturing drug products, including drug delivery nanoparticles, there are many factors needs to be considered. The first and foremost are the physical chemical properties. Among many important properties, particle size characterization is one of them, and is arguably the most important one, as they can significantly affect the stability of any colloid system and its in vivo and in vitro performance. So to be able to control the particle size and distribution of nanoparticles is critical. One more thing I'd like to point out here is that the particle size and distribution are also very important for generic drug development, because particle size and distribution is often a key criterion in demonstrating bioequivalence. The next requirement is that the selected technology should be robust enough and be able to produce repeatable results to ensure batch-to-batch consistency. The choice should also be future-proof in terms of scalability, which means the process should be easily scaled up for manufacturing with guaranteed results. Another requirement, or rather benefit, is the process should be able to improve or lead to easier downstream or adjacent processes. For example, injectable drugs requires sterilization to remove bacteria. Although autoclave is a widely used sterilization process, many active ingredients are heat labile, and that's prevent the use of high heat. A simple and more economic alternative way is to utilize stereo filtrations. The stereo filters are made of membranes, have rated pore size of 0.22 micron or 220 nanometers, which implies that the majority of the particles in the product should be smaller than this size to ensure an efficient filtration process. Last but not least, the manufacturing equipment needs to allow the entire manufacturing process to comply with regulatory requirements such as cGMP. Many pharmaceutical manufacturing processes also require the equipment is capable of running CIP and ACIP processes. Microfluidics technology can actually address all this kind of challenges and requirements from development to production with its unique advantages in achieving target processing results and guaranteed scalability. The principle of the microfluidizer technology and how the system works is illustrated here in this diagram. The product with large particles enters the system via the inlet and is put into a constant pressure pumping system, which pushes the material through a fixed geometry inaction chamber at pressure up to 30,000 psi or 2,000 bars, where the material experiences constant high shear rate and impact forces. All material will receive the exact same treatment to achieve consistent and reliable results. After passing through the inaction chamber, the product temperature can be efficiently controlled if needed. Microfluidics offer two different inaction chamber designs. As shown here, on the top is the Y-type chamber. The Y chamber has two microchannels, and the sample impinges on each other after passing through the channels. And this design is ideal for preparing most drug delivery nanoparticles such as nanoemulsions, liposomes, lipid nanoparticles, and etc. The bottom graph shows the Z-type chamber. The Z chamber only has one microchannel and is ideal for processing samples contained solid. Before I move on to the actual case studies, let me quickly explain how do we scale up and guarantee the results. Unlike other conventional technologies, when we scale up our system, we don't change the size of the channels. Instead, we use multiple parallel aligned microchannels with identical size. So as long as all other process parameters are kept the same, the product will experience identical uniform shear and impact treatment inside each channel, and that's achieve guaranteed linear scale-up results. This led to my first application, nanoemulsions. I will give an example of a drug-carrying nanoemulsion that was developed and produced with the microfluidizer technology. The aim of this study was to design a delivery system to carry widely used chemotherapeutics, toxins, and be able to deliver the drug to the tumor site. So the drug of interest here is a new generation of toxins developed with high potent and excellent efficacy to further reduce its toxicity and increase the retention in the tumor. The toxoid was also conjugated with DHA. So due to the lipophilic nature of the conjugate, oil in water emulsion is an ideal delivery platform to achieve desired efficacy and target deliver. The final emulsion would be ideally within the size range of 100 to 250 nanometers. To produce such emulsion, the researchers in this study used a microfluidizer processor and was able to successfully produce the nanoemulsions. As can be seen here, the resulting nanoemulsion had a unimodal distribution with an average particle size of around 200 nanometers and a polydispersity index or PDI around 0.1. So usually colloid system with a PDI below 0.2 is considered uniform. So in this case, it's well below 0.2, which means the produced nanoemulsion had a very narrow size distribution. The emulsion also had a desired negative surface charge to help prevent aggregation. Finally, encapsulating conjugate did not seem to affect the particle size at all. Analysis also confirmed that almost 100% of drug loading efficiency was achieved. The small particle size and narrow distribution also helped the stability of the produced nanoemulsions. As shown here in this graphs, the nanoemulsions were stable for at least a couple of months under all storage conditions. The best stability was obtained with storing at 4 degrees Celsius, which showed excellent stability during the 6 months duration of the analysis. Additionally, under all storage conditions, the nanoemulsion formulation was able to maintain the drug encapsulation level, while the free drug solution showed significant reduction and much faster speed. When intravenously administered, the nanoemulsion showed diffused pattern in naive mice and gradually decreased over time and almost cleared out within 24 hours. While nanoemulsion in tumor-bearing mice induced a massive signal in the periphery of tumor region and become more concentrated over the time and persisted over 24 hours. The plasma pharmacokinetic data showed that a nanoemulsion formulation significantly increased this plasma exposure with almost 6-fold increase in tumor-bearing mice, and the targeting efficiency was almost doubled with a nanoemulsion formulation. I'll move on to my next application, which is liposomes. Liposomes have been used as both drug delivery system and vaccine adjuvant in commercialized products. In this study, I'll be showing you some data from our collaboration with Professor Yvonne Paris' group at the University of Strathclyde. What we were interested here is another class of cancer drugs, Doxorubicin. Doxorubicin is a well-known anti-cancer drug, has been approved for treating several different kinds of cancer and tumors. Liposomal doxorubicin is formulated with pegylated lipid and was the first FDA-approved nano-drug delivery system based on pegylated liposome technology. It is currently available as Doxo in the US market. So the aim of this study was to develop a better process to produce generic version of doxorubicin liposome formulation with ease of manufacturability and scalability. So obviously, as mentioned before, the process needs to be able to achieve a target size range, which in this case is between 80 to 120 nanometers and with high drug encapsulation rate of over 90%. Another goal of this study was to see if the liposomes can be prepared without using any solvent. This is very important because so far, solvents such as chloroform or ethanol has to be used either sooner or later during the manufacturing process, regardless of the method of selection. The use of solvent is not preferred due to their potential safety and health concerns. So we want to see if the microfluidizer technology can be used to create liposomes without using any solvent. And we've actually successfully achieved this goal. So the new process illustrated in this diagram for making doxorubicin liposomes, you can see here, instead of forming lipid fumes as used in the conventional way, we simply hydrated all lipid ingredients directly in the aqueous buffer, and then use the microfluidizer technology, microfluidizer processor to reduce the vesicle size. In this case, empty liposome vesicles were created first. Doxorubicin was loaded into the aqueous core inside the liposomes via the remote loading technique afterwards. So to do this, a buffer exchange step is included after processing to create a pH gradient for drug loading. And finally, the liposome solution went through additional purification and sterile filtration steps. This is a busy slide, but the charts here show the particle size, PDI, surface charge, and drug loading profiles throughout the entire manufacturing process, shown in the previous slide. Results showed that the desired particle size and size distribution can be achieved by processing at 2 different pressures for just 2 to 3 passes. Both higher pressure and more passes resulted slightly smaller particle size and PDI. But particle size, PDI, and surface charge and encapsulation efficiency remained fairly constant throughout the entire manufacturing processes. So the reason you see a change here in the zeta potential is due to the change in the pH, which was required for drug loading, as I mentioned already. After loading, the net surface charge went from neutral to significantly negatively charged and stayed constant afterwards. We then compared the liposome created by the new solvent-free method to the conventional solvent-based method. So note here that although the microfluidizer processor can also make liposomes via the traditional way, for comparison purpose, an alternative method was used in this case, which was the sonication method. So you can see from the results here, the solvent-free method not only proved its capability, but also yielded overall better results. As the encapsulation efficiency was slightly higher compared to the solvent-based method. Clarity and images also revealed that the sonication method produced the non-uniform vesicles, whereas microfluidizer processor was able to create uniform small unilaminar vesicles. This confirms that the microfluidizer technology is not only suitable for producing liposomes, but also superior to other competitive technologies. Move on to my next application, which is lipid nanoparticle. These particles were developed to encapsulate and deliver nucleic acids such as DNAs and RNAs. Because naked DNA or RNA delivered by itself is rapidly degraded after injection, since under physiological conditions, those molecules are highly unstable. Therefore, proper delivery systems are required. Among several different approaches, lipid nanoparticles are by far one of the most attractive delivery platform. And the COVID-19 pandemic has also accelerated the progresses of this system even faster and made them the most successful system. In this study, lipid inorganic nanoparticles was developed for delivering replicon RNA-based COVID-19 vaccine candidate. So unlike the approach used by current RNA lipid nanoparticle-based COVID vaccines, researchers in this study took a different approach with two major differences. The first is the use of replicon RNA instead of the messenger RNA. Rep RNA provides increased antigen payload due to its replicative nature, which leads to more robust immune responses even after just a single dose, whereas mRNA typically requires multiple and orders of magnitude higher doses. So the second difference is how the RNA LNP system is prepared. Conventional approach produce mRNA encapsulated lipid nanoparticles within a single step. So this approach works. However, it has major issues of large amount of solvent and stability. It is believed that the RNA is the component that affects the overall stability. That's why both current mRNA LNP-based vaccines require extremely cold storage conditions, but still have very limited shelf life. The LNP themselves, on the other hand, are usually much more stable. That's why a two-step approach was used in this study, as illustrated here. The LNPs were manufactured without any payload, and the rep RNA was simply added just prior to immunization. As can be seen here, the resulting blank LNPs have a very small particle size of around 50 nanometers and a narrow distribution. After loading the rep RNA, the particle size grew slightly, which was an indication of successful RNA loading. Particle size monitoring of the blank lipid nanoparticles showed they were stable for at least 12 weeks, even at room temperature conditions. The RNA LNP complex also showed good stability as particle size remained constant for at least 2 weeks at either room temperature or refrigerated condition. In addition, rep RNA also remained its full integrity as confirmed by gel electrophoresis. Just want to quickly go through some of the results here regarding the immunogenicity of this vaccine formulation. So data show here that a single site immunization to mice with 10 or 1 micrograms of rep RNA lipid nanoparticle introduced a robust antibody response after 14 days. All 3 dose groups responded very well after 12 days of a booster immunization at day 28. So also all doses seems to induce Th1 biased immune response as indicated by higher IgG2 responses. Interferon gamma responses in spleens and lungs on 12 days after a booster immunization showed robust T cell responses for all those groups as well. So here the age-related mice model yielded very similar results, except at that the lower antibody responses were observed in the aged mice after the prime immunization. However, much higher antibody responses were developed after the booster shot for both doses. Finally, I'd like to point out here that although in this case study, the RNA LNP complex was used to formulate vaccines for infectious disease, The same concept can be used for cancer vaccines as well. But this approach is not just limited to vaccines. Using LNPs to deliver RNA-based drug also has a great potential in tumor therapeutics. Move on to my last application in this presentation. This is a very different system from all previous nanoparticles because it's a protein-based delivery system. Specifically, I'll be talking about albumin-based nanoparticles. Human serum albumin is the most abundant protein found in the human body, so it's biocompatible, biodegradable, and non-immunogenic. HSA also has multiple binding sites which can bind to either hydrophilic or hydrophobic therapeutic drugs. But most importantly, albumin can utilize the GP60 albumin receptor and spark protein pathway to effectively deliver albumin-bound therapeutics to tumor cells. All these advantages make SHA nanoparticle an ideal platform for delivering anti-cancer drugs that have high affinity for albumin. Picotexo, a well-known and widely used commercially available chemotherapeutic agent, is one such drug. The drug albumin complex is formed via the unique nanoparticle albumin bound or NAB Picotexo NAB technology. Abraxane is the first FDA-approved commercial product which is manufactured using the NAB Picotexo technology. So in this case study, the aim was to compare two different forms of NAB Picotexo. One is the non-cross-linked, which is similar to Abraxane, and the other is its cross-linked version. Non-cross-linked NAB Picotexo nanoparticle were prepared via the emulsion evaporation method. So basically, a Picotexo containing solvent is emulsified in albumin solution, which was done by the microfluidizer processor. After forming the desired nanodroplets, the solvent is subsequently removed to allow forming the nanoparticle albumin-bound Picotexo. The cross-linking version was obtained by adding additional cross-linking agent to the non-cross-linked nanoparticles. So the reason of doing this additional cross-linking step is some researchers believe this will help improve the stability of the nanoparticles. This can be seen from both particle size analysis and TEM images. The non-cross-linked NAB Picotexo nanoparticles were successfully produced with the microfluidizer technology with desired particle size and morphology similar to Abraxane. The mean size was around 130 nanometers with a very narrow distribution of PDI less than 0.1. Results showed that cross-linking had no effect on particle size and size distribution. Also, in both versions, very high drug encapsulation efficiency of over 95% were achieved. This slide shows the biodistribution of Picotexo and the anti-tumor efficacy. So you can see based on the AUC data here, the biodistribution of the cross-linked and the non-cross-linked nanoparticle formulations was slightly different. But more importantly here is the non-cross-linked nanoparticles rapidly accumulated into tumor, which indicated an enhanced tumor targeting efficacy compared to the cross-linked particles. This was also confirmed with the AUC data showed here that the non-cross-linked particle increased the percentage of drug distribution to the tumor by a factor of 2. So the enhanced targeting efficiency led to better therapeutic results as shown in this plot. Although both nanoparticle formulations could effectively inhibit tumor growth compared to the control group, the non-cross-linked formulation was more therapeutically active and had slowed down the tumor growth considerably. So I apologize that for the interest of time, I had to go through the examples rather quickly, but I hope from the 4 different applications I presented, you're able to learn more about the microfluidizer technology, and I'm convinced that this technology is the ideal solution for many pharmaceutical nanotechnology applications, especially cancer drug nanoparticle manufacturing. Before I conclude my presentation, I want to use the slides to quickly show you our diverse product lines. So the top are the lab units and the bottom are the pilot and production machines. So for this units shown here, all of our pilot and production models plus one lab models are all, we also have biopharma versions that are in compliance with cGMP requirements. And with that, I'd like to thank everyone again for your attention and be happy to answer any questions that you may have.